Hi everyone! Today we're starting a new chapter, chapter 21, which is all about nucleic acids and protein synthesis. We'll also be covering chapter 23, which is all about carbohydrate metabolism. So we're skipping chapter 22, but let's start with chapter 21. So to start with, I wanted to go over the central dogma of molecular biology. So this states that genetic information contained in DNA is transferred to something called RNA, and then that's expressed into proteins. So we have a nice little flowchart at the bottom that summarizes this whole process. So previously we talked about proteins and how they are very important for us to function as human beings. They're also really important in other animals as well. So proteins have many different functions, right? They can transfer molecules from one place to another. They can store molecules for use later. They can catalyze reactions so that they occur more quickly and more efficiently. And all of these different types of proteins make sure that we function day to day. So remember, we talked about the four different structures of proteins: primary, secondary, tertiary, and quaternary. And we said that the primary structure was the linear sequence of amino acids. But if proteins are built up from amino acids, how do we know what that sequence is? Where do we get that information from? Well, it actually comes from our DNA. So DNA has segments of what are called genes that contain information that are necessary for the synthesis of proteins. So each protein actually corresponds to a DNA gene. Now, DNA first has to go through something called RNA before the proteins are synthesized. So DNA transcribes its information into something called RNA, and then RNA translates its information into a protein. So this is the process that we're going to cover in this chapter. We'll start by going over the structure of DNA, how it works. Then we'll go over the structure of RNA. And then finally, we'll talk about transcription and translation and how proteins are synthesized. So let's start with the structure of DNA. So DNA is a double helix, and this is the secondary structure for DNA. So off to the right, you can see this double helix. It kind of looks like an alpha helix of a protein, but in a protein, there's only one strand that's curling in on itself. In DNA, there's two strands, and they're kind of curling around each other to make this double helix structure. So this structure was deduced by Watson and Crick, two scientists, in 1953. They actually received the Nobel Prize for their work, although their work was based on data from Rosalind Franklin as well, but she doesn't get credited as often with this structure of DNA. So this double helix has two intertwined chains. So you can see the blue chain and the purple chain there. So these are referred to as polynucleotide chains. So remember, when we talked about proteins, we said that the building blocks of proteins are amino acids. So the building blocks of DNA are nucleotides. So there's going to be multiple nucleotides in each chain, kind of similar to amino acids in proteins. Now, we're going to talk more about nucleotides in a bit, but each nucleotide has a base that sticks out, kind of similar to an amino acid side chain, right? So these bases are actually going to point towards each other in the center of our alpha helix or double helix. I'm getting proteins mixed up with DNA now. So these letters that are in the middle represent different nucleotide bases. And they're actually held together by hydrogen bonds. So these dotted lines are just hydrogen bonds here. So what I'll do here is I'll highlight bases in yellow, and then we've got our hydrogen bonds in purple. So these two strands are complementary to each other. They're kind of like puzzle pieces that fit together. So these bases actually hydrogen bond with only one other specific base. So these bases are called adenine, thymine, guanine, and cytosine. Adenine only bonds to thymine and vice versa, and guanine only bonds to cytosine and vice versa. So we'll talk more about the structures of these nucleotides. So RNA and DNA are both referred to as nucleic acids, and they're involved in the transfer of genetic information. So remember, DNA transcribes its information into RNA, and then RNA translates that information into a protein. So RNA's full name is ribonucleic acid, RNA. DNA's full name is deoxyribonucleic acid, DNA. And again, remember I mentioned earlier that the building blocks for DNA are called nucleotides, but these are also the building blocks for RNA. Now, an example nucleotide is shown below, and every nucleotide has three main components. A phosphate group, which is PO4, although sometimes there's a hydrogen there. A sugar, and that sugar is either ribose or deoxyribose. So ribose is present in RNA, deoxyribose is present in DNA, hence the two different names there. But we'll look at a little bit more at that structure in a second. And then finally, the last component is a nucleotide base. And remember, for DNA, we said that that was either adenine, cytosine, guanine, or thymine. I think I said all four. We'll talk about those too. All right, so just to summarize, nucleic acids are DNA or RNA, and those are made up of these nucleotides. And nucleotides have three components to them. They have a phosphate group, a sugar, which is either ribose or deoxyribose, and then they have a nucleotide base. So the nucleotide base is either going to be something called a pyrimidine or something called a purine. So we'll talk more about that in a bit as well. Okay, so first let's talk about those differences between ribose and deoxyribose. Okay, so ribose is a five-membered ring where oxygen is one of the members of that ring. And we've got our anomeric carbon at position 1. So remember, way back to our carbohydrate chapter, we said that the anomeric carbon was the one to the right of oxygen in the ring. So then we can just kind of go around the ring, number all of our carbons. Carbon 5 is that CH2OH group that's up to the upper left. So if we do the same thing for deoxyribose, everything is pretty much the same as ribose, except we're missing an OH group on carbon 2. So that's really the main difference between ribose and deoxyribose. We're missing that OH group on carbon 2 in deoxyribose. Now, phosphoric acid is just PO4. So phosphorus is in the middle, and then we've got 4 oxygens. And sometimes a hydrogen will be hanging out depending on the pH. And then finally, we've got that base, that nucleotide base. So in that flow chart, we saw that that could either be something called a pyrimidine or something called a purine. So a pyrimidine is a 6-membered ring. A purine is a 6-membered ring connected to a 5-membered ring. So in DNA, there are 2 pyrimidines and 2 purines. In RNA, we also have 2 pyrimidines and 2 purines, but they might be slightly different. So let's look at the nucleotide bases that are present in DNA versus RNA. Okay, so in DNA, which, let's see, I'll use the color red. So we've got thymine, cytosine, adenine, and guanine. So those are the 4 different nucleotide bases that exist in DNA. So notice that thymine and cytosine only have a 6-membered ring present. So they're considered pyrimidines. Now, there are some differences in the groups that are coming off of thymine versus cytosine, but they do both have that main pyrimidine structure in the middle. Adenine and guanine are both purines. So they have a 6-membered ring connected to a 5-membered ring. So they both have that same basic structure. However, some of the groups that are coming off of those rings are a little bit different, which is why these bases are different. Now in RNA, let's see, I'll use the color blue. So RNA has the same nucleotide bases as DNA except for thymine. So instead of thymine, RNA has something called uracil. So these are, that's the main difference between DNA and RNA when we're considering nucleotide bases. All right. So let's just kind of summarize. So DNA has thymine, cytosine, adenine, and guanine. RNA contains uracil, cytosine, adenine, and guanine. And we can just abbreviate those with a capital letter. All right. So how do we actually put all these 3 components together to make these nucleotides? So remember, each nucleotide has a phosphate group, a sugar, and a base. So what happens is the phosphate group is going to bond with the oxygen on the 5th carbon in the sugar. So here's our sugar, and it's 5 carbons. And then the oxygen that's on the 5th carbon is going to bond with the phosphorus in phosphate. So what we do here is we create a water molecule with OH and H, and then phosphorus bonds to oxygen. Similarly, the nucleotide base is going to interact with the anomeric carbon on the sugar, and we're also going to create a water molecule. So we end up with 2 water molecules, a bond between phosphorus and oxygen, and a bond between the nucleotide base and the anomeric carbon on the sugar. So this particular nucleotide that we formed is called adenosine for adenine, 5 for this 5th carbon here, monophosphate. So we only have 1 phosphate group attached to the oxygen on carbon 5. So we abbreviate this AMP. All right. And then remember, in DNA, we're missing that OH group on the sugar on carbon 2. So that's really important. And in the last slide, we actually had ribose, which does have that OH group on carbon 2. And that's for RNA. Okay, so again, the three basic components here, we have a phosphate group, a sugar, and a base. So let's review. Which nucleotide bases are present in DNA? And what are the three main components of nucleotides? So pause your video, see if you can remember this information. You can also go and look back at your notes. And then when you're done, we'll go over it together. Okay. Which nucleotide bases are present in DNA? All right. So the nucleotide bases are thymine, which we can abbreviate with a capital T, adenine, which we can abbreviate with a capital A, cytosine, capital C, and guanine, capital G. And then what are the three main components of nucleotides? So remember, we've got that phosphate group that's bonded to a sugar that's bonded to a nucleotide base. So the bases we just listed up above, what are the sugars? Which sugar would be in DNA? Deoxyribose. And which sugar is in RNA? Ribose. So you can kind of remember it just based on the first letter, right? Deoxyribose is in DNA, ribose is in RNA. Okay. So let's talk a little bit about that primary structure for DNA or RNA. So we already said that the secondary structure is a double helix, but the primary structure is the order of nucleotides. So this is very similar to those structures of proteins we talked about. So nucleic acids have a sugar-phosphate backbone, and it alternates sugar, phosphate, sugar, phosphate, sugar, phosphate. So remember when we saw the structure of our sugar, it was a 5-membered ring, and then we had that CH2OH group kind of up above. So when we number the carbons, we start at the anomeric carbon and work our way around. So we're going to put a little prime or apostrophe symbol next to each number. And what ends up happening is the 5' carbon of one nucleotide is actually going to join up with the 3' carbon on the next nucleotide. So if this is our next nucleotide here, so it will join with the 3' carbon on the next nucleotide. So we'll see a picture of that on the next slide. So the linkages between them are called phosphodiester bonds, and the order of the bases along the backbone will be unique depending on our DNA. All right, so here is that kind of backbone of our DNA or RNA, depending on which one we're looking at. Here we're looking at DNA, and I can tell because we're missing the OH group on the second carbon in the sugar. All right, so now notice if we start at the top, we're starting here with the 5' end of the sugar, and then that's going down to the 3' sugar, which is actually then connected to this phosphate group. And then that phosphate group, remember, is bonded to the 5' carbon on our sugar. We'll follow that down to 3', which is then connected to phosphate, and we can keep going down the chain. So this is why the backbone alternates between sugar, phosphate, sugar, phosphate, sugar, phosphate. So again, the link between that 3' carbon and the 5' carbon is called a phosphodiester linkage. Okay. Now, it's called a phosphodiester linkage because if we look at the bonds here, we have what looks like an ester group, but there's a phosphorus connecting them. So we've got two esters, but a phosphorus is what's connecting them. Okay. So if we were to zoom in on a section of our double helix, we would see that phosphorus, sugar, alternating backbone here. And notice that one strand is going from the 5' end up to the 3' end. The other strand is actually backwards. So it's starting at the 3' end and going up to the 5' end. So they're actually puzzle pieces, but they're kind of backwards. So one is flipped. Now, also notice that we have all of our nucleotide bases. So we've got adenine, thymine, guanine, and cytosine. And notice that adenine, A, is only bonded to thymine, T. So adenine only bonds to thymine and vice versa. Guanine only bonds to cytosine and vice versa. So we can only make AT bonds or TA bonds. And then we can only make GC or CG bonds in DNA. Also notice adenine is a purine. It has that 6-membered ring bonded to a 5-membered ring. And then thymine is a pyrimidine. And the same goes for guanine and cytosine. So guanine is a purine. Cytosine is a pyrimidine. So purines can only bond to pyrimidines and vice versa. But adenine, which is a purine, can only bond to the pyrimidine thymine in DNA. And the purine guanine can only bond to the pyrimidine cytosine in DNA. Now, the other thing to notice is the number of hydrogen bonds between each pair. So adenine and thymine can only have 2 hydrogen bonds, and that's just based on the groups that are coming off of each of them. However, when you look at guanine and cytosine, they can have 3 hydrogen bonds between them. And this is why adenine only bonds to thymine, because otherwise adenine wouldn't line up properly with cytosine. So adenine can make 2 hydrogen bonds with thymine. Guanine can make 3 hydrogen bonds with cytosine, but we can't mix them because they won't make the proper bonds otherwise. Okay, so AT and TA have 2 H bonds. GC and CG have 3 H bonds. All right. Now, I did find this really handy handout, I guess you'd call it, on compoundchem.com. This website has a lot of cool infographics on biology and chemistry, and one of them is on the chemical structure of DNA, and I think this really summarizes all of the information we've learned really well. So you'll notice the double helix in the middle, and it's color-coded with the nucleotide bases. So adenine is blue, thymine is yellow, guanine is green, cytosine is red. And they show you the nucleotide base structures there. It also shows you what we just saw, that adenine and thymine bond together because they make 2 hydrogen bonds with each other. Guanine and cytosine bond together because they can make 3 hydrogen bonds. And then you can also see the sugar phosphate backbone and how it connects. And then there's also a summary of how proteins are synthesized. So DNA is transcribed into RNA, which is then translated into a protein. Now, next, we're actually going to talk about this information on the bottom right. So we'll talk about how to make a protein from DNA. So we'll start getting into RNA and translation and transcription. So let's just review some of the info we learned. So adenine and guanine are blank. And what I'm looking for here is either purines or pyrimidines. Thymine and cytosine are blank, so either purines or pyrimidines. And then which 2 pair together? Specifically, blank pairs with blank and blank pairs with blank. So which nucleotide bases pair together? And then the type of bond that holds the nucleic acids together is blank. Or sorry, that should be nucleotides, not nucleic acids. All right, so see if you can complete these sentences. If you get a little confused, that's okay. We'll go over it together in a bit. All right. So let's go over each of these. So let's talk about adenine and guanine. So are adenine and guanine purines or pyrimidines? Purines. They have that 6 and 5 membered rings that are bonded together. Thymine and cytosine are pyrimidines. So they just have a 6-membered ring. So purines and pyrimidines always pair together on the structure of DNA. Specifically, adenine pairs with thymine and guanine pairs with cytosine. And it's okay if you mix those up, if you put guanine and cytosine first. And I just want to make sure you know what pairs with what. All right, so the type of bond that holds the nucleotides together is the hydrogen bond. And how many hydrogen bonds exist between adenine and thymine? 2. And how many hydrogen bonds exist between guanine and cytosine? 3. All right, so we learned a lot of information today in this video specifically, but let's just do one more problem. Okay, so let's say that one strand of a DNA molecule has the base sequence C, C, A, T, T, G. What would be the sequence for the complementary strand? And remember, cytosine and guanine pair together, and adenine and thymine pair together. So what would the other strand look like in order to create those hydrogen bonds? So pause the video, see if you can find the matching pair for each of those nucleotides, and then we'll go over it together. Okay, so we said cytosine can only pair with guanine, right? So we'll just put G and G across from cytosine. We said that adenine can only pair with thymine. So we'll put a T across from A. We said that, again, thymine can only pair with adenine. So we'll put A's across from the T's. And then G, what pairs with G? Cytosine, C. All right. So typically what we do is we write our strands going from the 5' carbon to the 3' carbon. So that's our first strand. And we know that the complementary strand has to be the opposite of that, right? We saw that earlier. So the opposite strand, or the complementary strand, its 5' will be on the right, and then the 3' carbon will be on the left. So typically we start going from 5' to 3', and then the complementary strand will be the opposite. So just some tidbits of information there. Okay, so next time we're going to talk more about the process of forming a protein, starting with DNA, going to RNA, and then finally our protein. So I will see you then.